In the human body, we have veins where our blood can flow through our bodies. Plants have veins that water flows through. The plants suck up the water from the dirt and the water flows through the plant. So how can we tell if the water is going through the plant? Okay, in vascular plants, there are tissues inside the plant called xylem and phloem. And as we learned from our notes in class, xylem takes water and nutrients up the plant, up towards the stem and the leaves and the flowers. Remember, xy to the sky. So what we have here with our Xylem lab is I have some celery. Remember, celery is a plant. I also have some daisies today. I have a cup that has water in it. This is going to be our control cup, okay? So the water is going to remain just plain water. Then I have another cup that I've labeled food coloring cup. I've also labeled another cup with salt water. So I'm going to go ahead and prep our lab, our Xylem lab, and I'm going to go ahead and take a piece of the celery and I'm going to place it in our control cup. We need to make sure we have a control cup and that nothing changes with the water, that it's just plain water, so we can see if anything changes with our piece of celery. We want to make sure that the leaves don't just turn color on their own. The next cup we have is our food coloring cup. And right now there is just water in this cup, but I'm going to go ahead and add some red food coloring to this cup. I'm going to put in probably about 6 drops of food coloring. I'm going to go ahead and stir that up. You can see now the water is red. I think I want to put a few more drops in, 7, 8, 9, 10. Okay, so I put 10 drops of the red food coloring. You can see it made the water even brighter. Okay, so I'm going to go ahead, stir that up, and go ahead and put another piece of celery into our cup with water and food coloring. The third cup, I'm going to use my piece of celery, and I'm going to add some salt to this cup. And I'm going to just pour some salt in there, and again, I'm going to go ahead and stir that up so that the salt's not just sitting at the bottom of the cup. So now we have a nice salt water solution that we can put our third piece of celery into. And being that this is the Xylem lab, of course we are trying to see if xylem is present in these pieces of celery. Remember, xylem in plants brings water and nutrients up the plant, xy to the sky. Okay, so that is my third cup with salt water, my second cup water and food coloring, and then of course that's my control cup. Nothing is in that cup except water. We need to make sure that our celery doesn't change just by being in a cup of water. Now, I'm going to push these back. I'm going to also today have a little fun here. See if xylem is present in my daisy flowers. And so I have 3 different flowers. I'm going to go ahead and we'll try blue food coloring. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And our first cup, I'm going to stir that up. So now we have a cup that just has plain water and some blue food coloring in it. I'm going to go ahead and put this one daisy in the blue cup. I'm going to make another red cup. Again, this is just water with red food coloring. And just for fun, I'm going to go ahead and prepare another cup with just plain water. Here's my plain water cup. I think this time I'm going to go ahead and grab some green. I'm going to go ahead and stir that up in that cup. And now this is just, again, plain water with green food coloring. Okay. So I have a bunch of different cups. I have celery in just our control cup, which is just water, celery in food colored in water cup, celery in a salt water solution, so it's water and salt. Then I went ahead and we're going to have some fun with these daisies. And I have blue food coloring in water, red food coloring in water, and green food coloring in water. We are going to wait 24 hours, that's one day, to look at our results and make observations at that time. However, what you have is this worksheet that we need to start the first portion of our Xylem lab, okay? Right here up at the top, it says hypothesis. And there's two different questions that you have to answer with your hypothesis. I wanted to share this with you guys. A hypothesis is a prediction or proposed explanation made with limited evidence as a starting point for further investigation. So it's a prediction of what you think might happen. So on the first hypothesis, it asks us, what do you think will happen to a celery stalk if it is placed in a beaker with water and food coloring? That's a prediction that you need to make. So your hypothesis is a prediction of what you think is going to happen when you put that celery into a beaker with water and food coloring. On B, what do you think will happen to a celery stalk if it is placed in a beaker with salt water? So again, you're making another prediction of what you think it will look like, the celery will look like if you place it in a beaker with salt water. Again, we don't know because we don't have the evidence yet. We will come back in 24 hours and we will look at our evidence and make observations. Okay, so now it's time to go ahead and fill out and complete our Xylem Lab worksheet. We're going to go ahead and move to the bottom where it says Xylem Lab data table. And we are going to look here in the first column. It says Beaker A. That's going to be our cup that we put the celery inside of, where it only had water inside of it. And I'm going to show you here. This is our control cup. It's only water. There is a piece of celery in it, a celery stalk with some leaves at the top. Again, we put that celery into the control cup so that we can make sure that the celery doesn't change colors or something else doesn't happen to it. So again, it's just plain water. And again, it looks like this. So I need to draw this out on my lab worksheet. So I am going to go ahead, and it says to be detailed. So there is my water. I have some colored pencils as well that I will be using today. I'm going to go ahead and give it a blue color. Because it's just plain water. And of course I have my celery stalk and I have some leaves on the top to show my celery. Okay, so these are the observations that I made on the first beaker, which is a plastic cup. It's our control cup. It's just plain water with celery in it. That is before. That's how we set up the lab. Okay, so of course we're going to wait 24 hours, make some new observations to see if anything happened with that celery. All right, so it says down here in your words, write down your observations. This is the before cup, and I'm just going to write down plain water in cup with a, let's just put green celery stalk. Now you can write in your own words the observations that you made from that picture. Okay, now we're going to look at the second cup, and what we have is a setup with a celery stalk again inside a cup. This is the cup that we added the red food coloring to. So make your observations at this time. Take a look at that cup and think about how you're going to draw that out on your Xylem Lab worksheet inside that first beaker. Okay, this is the prior, the beginning of the experiment, the investigation. We have not let it sit for 24 hours. This is just our beginning image of red food coloring and celery in a cup. Okay, so we're going to go ahead and look at our lab sheet, and now I need to draw this out. I'm going to go ahead and get a red matte pencil. This way I can color whoops, the water red, okay? And again, I come in with my green to draw my celery stalk. Okay, so it did say to be detailed in your lab, so that's why I'm using color, so I know what it looks like. Okay, remember we're doing a before and after, but we have to wait for 24 hours to make new observations. Okay, this is the investigation. And again, it's called the Xylem lab, so we're investigating whether xylem is present. And again, you're going to write down here in this box right below your picture of the celery in the food coloring, in your words, write down your observations. Okay, and I'm just going to write down my observations. Place celery stalk in water and red food coloring. Okay, it's in your words, so you can write your own observations. Okay, so now we need to take a look at our third setup. And what we have is we have a cup with salt water and a piece of celery inside that salt water. And again, this is the beginning setup. We will make observations in 24 hours to see if anything has changed. But for now, I need to complete my Xylem Lab worksheet. So I'm going to go ahead and add my last entry. And I'm going to go ahead, I'm going to use gray on this one just because there's salt inside that water. And again, I'm being detailed with color so that I know what it looks like. I'm making an observation. Making an observation is just taking a look at it, see what's going on. What does it look like? Okay, so here is my celery plant in salt water. That's what the cup looks like. Of course, in my own words, I need to write down here what my observations are. And so I'm just going to write down that it's a piece of celery inside a cup of salt water. All right, so this is going to be fun. We're going to wait 24 hours and then we'll take a look and see what happened. Again, this is called the Xylem Lab. We'll see you in 24 hours.